Hello, everyone. Good day, everyone. This is Mr. Lee, and today we will be talking about cell division. And for your information, there are two types of cell division. One is mitosis, another one is meiosis. So for today's lesson, we'll be only talking about mitosis because it's already a very complicated process. And before we go into meiosis, you have to master the concept of mitosis. All right, without further ado, let's go to the next slide. Okay, so before we go into all the details of mitosis, you have to first understand what are somatic cells and what are gametes. Okay, because mitosis only happens in somatic cells, while meiosis happens in gametes. So what are their differences? So for somatic cells, they are all the cells in your body, from your skin to your liver to your eyeballs to your muscle, blood, gut, heart, and so on. All the cells in your body except the sex cells, okay, which are the egg and the sperm, okay. Okay, beside these two, all the other cells in your body, they are all somatic cells, and mitosis happens in them. Okay, what about gametes? Gametes, they only meiosis happens in gametes. So, as I mentioned just now, mitosis is the division of somatic cells. All the cells in your body other than the sex cells. So all of them are called somatic cells, and they divide into two daughter cells. That's basically mitosis. And this is the cell cycle. Okay, the cell cycle is basically the life of a cell. Okay, what it has to go throughout its life, and it has four phases here. And the first one, which is the pink one here, is called the G1 phase. G stands for growth. 1 stands for the first growth phase. It takes up a big chunk of the cell cycle, and it goes to the second phase, which is the S phase. The S actually stands for DNA synthesis. Synthesis means making, the production of DNA, or to be more precise, is actually the replication of DNA. Okay, the copying of DNA happens in the orange part here of the cell cycle. Then you go to G2, the second growth phase, as it prepares for mitosis. And finally, we come to mitosis, which only takes up a small chunk, a small part of the cell cycle. Okay, this cell division happens here. Okay, in this next slide, the diagram here is more detailed. So as you can see, the G1 phase, which we mentioned just now, happens from here all the way to here, then S phase, and then G2 phase. So these three makes up the interphase. As you can see in the inner ring here, there's a name for it. It's called the interphase. And other than the interphase will be the mitosis phase. And do you know what? Mitosis itself also has four phases, which is where's my cursor? Okay, which is the prophase, metaphase, anaphase, and telophase. P-mat. Okay, you need a proper mat in your living room. That's how I remember. P-mat, a proper mat. Okay, so just now that diagram is a little bit complicated, so this is a simplified one. So from this diagram, you can see that The red part of the cell cycle is the interphase, which comprise of the G1S and the G2 phase. Then the brown part here is the mitosis phase. And as I said just now, mitosis itself has the pro, meta, ana, and telophase. Not forgetting the cytokinesis phase. Cytokinesis meaning cell division. Cyto means cell. Kinesis means dividing. All right, so from that diagram there, and from now on, I will be explaining each phase in detail. And in all of these slides, I will put a small diagram here on the top left corner, so that we can keep track of what's going on, where we are in mitosis. So now we are in the first phase of mitosis, which is called the prophase. Pro actually is it's a Greek word for before. Okay, so in prophase, and yeah, for every phase I will have 2 slides, and one is simpler like this, and the next one it will be a bit more sophisticated. And in prophase, okay, so let's come back to this slide here. So in prophase, the chromatin is actually long thin threads of chromosomes, and it will condense. Okay, it will condense, which means it will thicken. Okay, it will become thick, and they will condense into chromosomes inside this nucleus. And in the nucleus, there's one small organelle called nucleolus. Nucleolus, as you have studied in Unit 2. Okay, so nucleolus will disappear. And what about these yellow things? Okay, these are centrioles. The whole thing here is called the centrosome. And inside of each centrosome, you have 2 centrosomes. In each of the centrosomes, you have 2 centrioles. Okay, so we go into detail now. This is the cell, and nucleolus disappears. The nucleus is in the middle of the cell here. The nucleolus disappears. The chromatin condenses into chromosomes, as you can see, forming the X shape. Okay, the formation of spindle fibers at the centrioles. The blue lines are spindle fibers, and the centrioles will move to opposite poles. Okay, and now we have come to the second phase of mitosis, which is the metaphase. So in metaphase, the chromosomes will line up along the metaphase plane. As you can see here in the middle, it forms a metaphase plate. It is also called the equatorial plane or the imaginary plane in the middle. So this is the more sophisticated diagram. It shows that the chromosomes are all lining up aligned on the metaphase plate, and each chromosome, consisting of 2 sister chromatids, is attached to the spindle fiber, the long blue line here, by the centromere. So in the middle of this chromosome, the whole X shape here is called a chromosome, and in the middle there's the centromere. Okay, the centromere is attached to this spindle fiber. And what about sister chromatids? What are they? So sister chromatids is actually the one side of the chromosome, and there's 2, and they are tied in the middle by the centromere. You can imagine it as 2 sausages tied in the middle by a rubber band. So the rubber band will be the centromere, and the 2 sausages will be the sister chromatids. Okay, so each chromosome has 2 sausages, 2 sister chromatids. Okay, and it's tied in the middle. So what happens next is the next phase is anaphase, the third phase. So in anaphase, these chromosomes will break at the centromeres in the middle, and the sister chromatids will be pulled apart, okay, and they move to the opposite ends of the cell. Okay, sister chromatids. So in this more detailed diagram, The spindle fibers connected to the centromeres, connected to the sister chromatids, they will shorten, thus pulling the sister chromatids towards the opposite poles, as you can see here. Okay. What about the unattached microtubules? What about those that are not attached to centromeres? They will push against each other to elongate the cells, to make the cell longer. As you can see here, 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 and here. Okay. And finally, we have come to telophase, the second last stage, and of course cytokinesis later. So the nuclear membrane will now reform in each of the daughter cell, and the nucleoli, which is the plural of nucleolus, will reappear. And of course, the chromosomes will now unwind back into chromatin. So in this diagram here, as you can see, the new nuclear envelope will slowly reform, and the chromosomes will unfold back into chromatin. And of course, the nucleolus, the nucleoli, two nucleoli will reappear, and the cell continue to elongate in the telophase. Last but not least, we have come to cytokinesis, the cell division stage, where the cell from one cell divides into two daughter cells. So this is cytokinesis. So all in all, in conclusion, this is a summary of mitosis. So from the prophase to the prometaphase to metaphase, as you can see here, the chromosomes are aligned in the middle on the equatorial plane. Then in anaphase, they will be pulled apart. In telophase, actually this diagram isn't that accurate. You should be able to slowly see the formation of the new nucleus. And finally, in cytokinesis, two daughter cells will be formed. So that's basically mitosis. And the process of meiosis will be explained in another video, because otherwise this video will be too long. So thank you for watching. I hope you have become clearer in the concepts, in all the stages, all the steps in mitosis. Okay. And if you have any questions, any comments, any suggestions, please leave it in the comment below. Thank you very much, and I'll see you in the next video. Thank you.